From Microbe TV, this is Office Hours for Wednesday, the 29th of January, 2025. I'm Vincent Racaniello, and welcome to your corner, your viral corner of the internets. And welcome our moderators tonight. Les is here. Vanity Nutrition is here. Tom Steinberg is here. Thank you for joining us and keeping it civilized. Tell us where you're from. Frank is here tonight. Hello, Frank. Tell us where you're from. We'll shout you out. My plan tonight is, we're going to talk about RFK Jr. I didn't get a chance to see, so I want you to tell me what happened, and we'll chat about it. And we don't want to dwell on that too much. We want to dwell on the science we love, so we'll have a mini lecture as well. So Wednesdays are tough now because I teach at Columbia at 4:10 to 5:25. Then I hop on the number 1, the Penn Station, catch the 601, which is very tight. Change trains in Newark. I just got home 15 minutes ago. I grabbed a bite to eat, and here I am. That's a bit exhausting. But I'm happy to do it. I like doing this very much, as you know. I Am Not Drunk is from Queen Siriket Park in Bangkok, 22C, with flowers in bloom. Here it's cold. Let's see what the temperature is these days. -3 Celsius, -5 Celsius. It's going to be cold all week, all below 0. It really hurts. Patricia is from Massachusetts. Wayne is from Urbana, Illinois, 34F, 1 degree C. Dagfin is in Oslo. Fascinated by the hearing. RFK is truly unfit. Yep. Tatiana Beast Mode is in Birmingham, 57F, 14C. Abdul Rahman, 4 degrees in Brooklyn. Costello says, I wish this were on Twitch. I can put it on Twitch too. I can dual stream if you want. It's easy to do. I know you mentioned this last time. It's just a matter of me pushing a button. Patricia's in balmy DC. Here's Tom, our moderator, one of our moderators. I'm back in Western Oregon after a trip to Madison, sitting out a COVID-19 exposure. Pat is in Los Angeles, 56F, no smoke. Ray's in Sacramento, 14C. And there's Frank. Hello, nice to see you. It's been a while. Will is in Changsha, China, 5 degrees C and hazy sunshine. Well, America is currently a S show. I give thanks for Vincent, etc., who give us some hope. Well, thank you. I can't take a lot of credit, but I do my best. We do our best. I have a lot of people at Microbe TV that feel the same way, right? Here's Carol Wong, our favorite nephrologist from Bakersfield, 58F, 14C. Vanity Nutrition is at JFK. Got an invite from Columbia for Dr. Fauci's fireside chat. I won't. I wasn't invited. You're a former student, so you got invited. It's good. Visto tutti. Buonasera, visto. 23C in Sydney. I mean, I think this would be fun to listen to this Fauci thing. Did you hear? This is disgusting. Trump took away his security detail, and he said, I won't feel bad if something happens to him. How is this not the nastiest person on earth? And you really think someone like that is going to care about you? Are you delusional? Oh my God. Wouldn't feel bad if something happened to him. Hello, Bernard. Chevy Chase, Maryland. Privyet Veronika from Moscow. It's 4 degrees C, but it's bad because there should be snow. Plants need their beauty sleep, and birds shouldn't be coming back yet. Chris Lepowski. Really cold near Montreal. It was interesting to watch the RFK confirmation hearings. What's your opinion? I didn't watch. I was really occupied with other things. But I would like you guys to tell me about it. Dean is in Wisconsin, 32F. Patricia's in Massachusetts. Mark Martin is in Tacoma. Not very cold and no snow. Nebby is in Central Florida where it's 65 degrees F. Yeah, discophage. It's discophage. I left it on overnight one night by mistake, but it's still going strong. Those little batteries. It's cool, isn't it? I love it. Thank you. Thank you very much, Mark. Maybe RFK should figure out how government pronouncements nearly give people heart attacks. Well, at least Trump rescinded the grant cuts because it turns out it breaks a law. You can't do it, and the judge was already serving it, and he didn't want to lose in court. So that's good. That's good. Dennis is here from Nebraska. Elizabeth is in West Virginia. Patty's in Massachusetts. Angel is a third-year vet student from Georgia. Elizabeth ripped him a new one, yeah. So she wrote a great letter that's online. You can find it to Congress. Basically, she said, you're not qualified at all. You have no background. It's crazy for you to even be considered for this. Logotic, 4 degrees C in Chicago. Julie S. is in cold, windy North Central. David is in Dayton. Questella has never been on a bus or train in her life. You can avoid a lot of infections that way, Do not burn out. I won't burn out. When I was in high school, my student friends used to say, Racaniello, you're going to burn out before you're 30. And I never did. I never felt I was burnt. I really liked what I found to do, so that's the answer. There are days when I'm tired. I just take naps. Well, at the incubator, there's a little sofa. John is in Minneapolis, 36F2C. Julie F. is in North Central Pennsylvania. Tona is in Long Island. Frank is currently battling COVID. Very miserable. Fever won't break. Extremely tired. What a huge difference from 2023. You're probably not taking Paxlovid, right? I'm sorry. Yeah, it can be miserable, but it will go away. Anatomy and physiology tutor is from Vancouver, BC. It's minus 3C. EHS Talent is in Lehigh Valley, Pennsylvania. Jocelyn is in Sarasota, where it's been in the 70s all day. David saw Paul Offit on CNN tonight, missed first half. So Daniel Griffin was with David Learer live commenting on the hearings, I believe. I heard. I didn't get to see that either. Mark Martin will be in New York in late February and able to see the incubator, maybe attend a lecture. If you want to, it's fine. It would be fun. Eliza, 6 degrees in southern Ontario. Betty K., your loyal listener from Washington Heights, after today's RFK hearings, hiding under a blanket. I am not drunk is exactly what a drunk person would say. We have 160 people. That's great. Hit the like button, folks. Get some other people here. Learn some science. Hans, hello from Guayaquil, Ecuador, 29C. That's so nice and warm. I could really use it. So I have a lovely thermal turtleneck. It's very warm, and I got a sweater on top. It's perfect. I don't want to have to do that. I want to sit in the sun and feel good. I remember doing that in August. Dave is in Georgia. Lise is in Ohio, of course. Tracy's in Chapel Hill, where it's 56F13C. Nevitz, the RFK Jr. hearings were bonkers. Too many people have been mesmerized by the Dr. Campbells of the world. He's such a bad person, too. Eliza, minus 16, clear in southern Ontario. Greg is in New Brunswick, Canada, minus 13C. There you go. Silent Runner, 53F in Wichita, Kansas. Felt like spring today. John writes, some senators used visuals, and I thought the best one was Sanders with the anti-vaccine shirt sold from his own firm, which he tried to deflect from. RFK Jr. is suddenly a born-again pro-vaccine. Who knew? Is that what he said? You shouldn't believe him, of course. Laura is in Washington, Pennsylvania, where it's 3C. Yoda, minus 20, with a minus 27C wind chill in eastern Ontario, bracing for a trade war. Love your show. Frank is in Santa Barbara. Paul Offit was straight to the point and packing in the punches during last week. Bernie Sanders hearing. Yes, that was very good. Jill writes, hi, all. Sending deep breaths and or stiff drinks to everyone from Wilmington, Delaware. Well, I just have some ginger ale here. And it's gone, but it's just very refreshing, and it helps it helps an upset stomach. So I like to drink that, but it's very sweet, unfortunately. Can't have too many. Kip and Laura, thank you for your contribution to science communication. Can't believe we're not going to get MMWR and NIH pubs. Yeah, it's terrible. Emerging infectious diseases. It's ridiculous. Patricia says, now I'm thinking the only answer is to be as mean and obnoxious right back at them. I'd love to do it. So liberating to ream out someone who's mean and ignorant and just tell it like it is. Yeah. Well, the Democrats don't get mean. That's the problem, I think. Mike and Lori, 42F in Seattle. Spamlet is making bread in a hot kitchen in Bedfordshire. Sounds cool. Hello, Midge. Nice to see you again. EHS says RFK's a liar like many other politicians. I was furious. I just watched the BBC clips of RFK Jr. He just lied through his teeth. That's what he's been doing for 20 years. Lying through his teeth. He refuses to acknowledge all the evidence that indicates his views of things are wrong. He completely refuses and he sticks to his stupid theories. And people believe him. It's crazy. Marie, thank you for all you do to keep us healthy. I don't know about healthy, but certainly informed, right? None of them care about public health, heart attacks, or anything else. This is absolutely correct. Aspie Third Eye, I do clerical work at UTHC at SC. Memphis offices were freaking out today over the funding freeze, which now is lifted, I hear, right? Medicaid portal remains closed. I don't know why they felt it necessary to silence all the communications, right? They say, oh, we want to make sure everything that's being communicated is consistent with our views. No, that's not how science works. They're not going to be consistent with your views. Well, first of all, you don't know anything. Nobody in the administration knows any science. So to say consistent with our views, and anyway, there are tens of thousands of grants. There's no way you can go through those and decide, right? It's just a nonsense. It's a power play. Lee says RFK Jr. perjured himself during his testimony per the latest news. What did he do, Lise? Tell us. Hat says impression is that he's a con man and a liar, a GD liar. All lawyering techniques do not work in science. He never took a single science class in college. Yeah, but he sees that he can make money in science, and so that's why he's doing this, right? Yep. Elizabeth says apparently the press secretary said the funding was still on hold. They just canceled the memo. Probably still going to be a lawsuit. There should. Everything they do should be sued. Not give them any easy out. Abilash, good morning. 5C in Frankfurt, Germany. Ripped comp. Taking your course for the 5th year. Yes, so you're now, it is the 17th year I'm teaching this virology course. It's a good crowd this year. It's a packed room. Looking forward to it. Spamlet writes, nobody said a word about WHO at the RFK Jr. questioning. Yeah, you should have asked him what's his opinion to that. Yeah. Steph SF, welcome, our moderator. Good to see you. Gracie writes, from what I learned on Twin, nothing RFK Jr. can be trusted, says can be trusted. Once the brain is wired to lie, it takes a conscious effort not to lie. He took no breath between their questions and his answers. And he's not thoughtful at all. He's just got rote lies in there and they're just pouring out. You're absolutely right. John is in Port Townsend, Washington, 40F. Welcome, John. Polka Dot says Dr. Offit did a great job on PBS tonight. Got straight into the point. It's great. Good to hear it. Frank was prescribed Paxlovid, but is chicken to take it. No, it's fine. It's just a bad taste, that's all. So don't be afraid. Markle, thankful it got up to 45F on the east end of Long Island today to enable me to get the last of the icy snow off the shady part of my driveway. Les loves the Beyond the Noise questions for JFK. Thanks. I'm glad you like that. Offit is good about that. Yep. A lot of people are telling Frank to take the Paxlovid. It helped Frank a lot. It helped Aspie a lot. So, Frank didn't know anyone besides me who took it. Well, Dixon has taken it. Who else? I think Daniel has taken it. Artemis, is the incubation time of the current circulating strain of SARS-CoV-2 virus? No. I think it's the same as all the others. This idea that this incubation period is changing is, in my opinion, incorrect. It's very difficult to do that. It's up to 7 days. Patricia took it twice. Vincent took it. Lots here have taken it. After the taste, it goes away. Totally worth it to me. Leo, hello. 47F in Mendocino. Cool. Patricia says there's no rebound. Rad is in Toronto. -5. My hands are cold. Get some gloves on, no? Mike wants to know, is there a common cause between Pentagon ran a secret anti-vax campaign to undermine China and recent lab leak promotion? No, I don't think so. I think the recent lab leak promotion is because we have a change in administration that did not like the previous CIA, FBI conclusion of, with low confidence. We think it came from a lab with no evidence. So they wanted to erase that doubt, which is just not based on any science. It's disgusting. Really is disgusting. Tona says, anyone else here off it? When asked if he'd approve of RFK off it, shrieked, hell no. Yeah. Rebound is part of the normal course of the illness. It's nothing to do with Paxlovid, but so many people insist because it happens with Paxlovid, but they don't understand it also happens without. March 7/16 is in Central Virginia. Clear in 10 degrees C. All right, Frank is going to take Paxlovid tomorrow and report next time. Okay. Hello, Rita in southwest Idaho. Sunny and no wind, 32F. That's pretty nice. Hello, MK. Eastern Massachusetts, 32F. See you later. Yeah, I know a lot of funding panic has been happening all over the U.S. It's not good. You have to make people scared. Is that your life's goal? It makes you happy? Ridiculous. Vista wants to know if infectious disease information will flow from USA to WHO. No way. If you look at the memo, it says no communication, no information flow to WHO. We're going to make our own mini WHO within the U.S. Good luck. Good luck with that. Lori is in Charlottesville, Virginia. Welcome, Lori. Christella, thank you for your support of science communication. Lisa wants to know if Australia will get flu strain information from the U.S. Yeah, for sure. We will share it with other countries. Well, I would assume for sure, but you never know. But normally that would come from WHO, right? Will says, thank you for breaking down Pull Off It's Beyond the Noise videos into shorts. Many will not watch the full episodes. You're welcome. We thought it was important. Nevitz wants to hear Jesse Bloom. He's been on Twiv a couple times, actually. You can go search for it. Yep. Midge says, informed equals healthy. That's so true. It is so true. I can't see how anyone could vote for approving RFK after today's hearing, but I'm afraid they'll vote for him anyway. I'm scared. Yeah, because they say we will give Trump what he wants, which is, it's so, it's a word. It's, why do you have to do that? Well, he'll retaliate if you don't. This is the era of retaliation, not collaboration. And that, more than anything, I find objectionable that many Americans vote for a person who is like that, just mean and retaliatory. Mike says RFK Jr. only got into environmental activism as community service for his heroin drug charges. He loved needles when they contained junk, went from junk to junk science. Yeah. Science is not about views or opinions or wishful thinking, period. It's quite the opposite. Yeah, it's not about your view. We don't care what your view is. RFK Jr. says vaccines don't work. I don't care if they do. You're just wrong. But unfortunately, he may be in a position of power, and that's really bad. He wants to change vaccine schedules. It's a good article in Science-Based Medicine. I forgot the author, but he's very well written about this Danish physician kind of coming out in favor of RFK and just getting it all wrong, getting all this facts. He says you don't need to immunize against Hep B, and he's totally wrong. And this guy puts the numbers out there, and it's very, very persuasive. Theoretically, liposomes with mRNA can be used to transfect nasal cells. Yes, people are trying it. RFK is a rubber stamping numbskull. Marco, I thought it was eye-opening of Senator Warren to reveal how RFK Jr. makes millions from directing people to a certain law firm for lawsuits against vaccine companies using his name and expertise. Also, he's involved in this lawsuit against Merck, against HPV vaccine. And he's going to keep any money that he gets from it. It's totally conflicted. Truman is in Fort Worth, pouring rain and thunder, 58F. Well, Will, as bad as RFK Jr. etc. is, the GOP senators are worse for approving obvious lies and liars. Yes, just to appease Trump, it's horrible. They're just supporting lies. Dagfin, how can RFK claim he will adhere to gold standard of reproducible science when he both denies and ignores it and doesn't even understand it? He keeps lying. Yes, everything is a lie. Whatever he says, don't believe it. It's going to be a lie. Heather is in Vancouver. Welcome. Penelope wants to know, are there over-the-counter flu tests? Yes, there are. No OTC medicine, though. Well, the stuff that really doesn't work. The prescription drugs, Tamiflu, Baloxavir, you got to get scripts for that. Eliza watched your first lecture. It was great. Thank you, Eliza. Glad you like it. Les said he majored in opioids in college, so he has pharmaceutical expertise. Yeah, Ripticom would have loved to see Amy grill RFK Jr. That would be fun. Luckily, scientists are not a dime a dozen. Their offices may not be allowed to officially interact, but people do and will. Sounds like Vincent is, hello, Star Climber. Sounds like Vincent is rocking a gnarly cold. And RFK Jr., mendacity, and Kennedy format is still gross dishonesty. Yeah, I have a bit of a cold. My second one of January. I'm just getting over it, but really takes a lot out of me. Yeah, this is a good question, Lillian. If he doesn't get the nomination, I wonder who'll be the next choice. Maybe even worse, although maybe you can't get anyone worse. But it will be some objectionable person with contrarian views. Less circus. You know, there's a Turkish proverb someone sent me today. See if I can remember it. When the clown moves into the palace, he does not become a king. The palace becomes a circus. Polka Dot got her pneumonia vaccine today before they removed funding. Rita took Paxlovid and COVID symptoms were gone. Yeah, mine were gone in a day also. If I had Stanley Plotkin on, no, I really want to. I'm very behind in all the things I want to get on these programs, but he's a tough one, yes. Happy Lunar New Year's. Hello, Jessica from the University of Toledo 31F in Ohio. Yes, Truman took Paxlovid twice, 0 issues. As you can see, a lot of people have good experience with Paxlovid. Polka Dot, Pup. I heard the government had withdrawn funding for AIDS in Africa. Yeah, they withdrew funding for PrEP, pre-exposure prophylaxis. So the U.S. used to give drug to Africa and many other countries to the tune of $7.5 billion a year. They suspended it for like 150 days. So people are going to get infected. It's a death sentence. Yes, brilliant. You're right. John says, I find the 2025 virology course dusting off my memory. Glad I have answered the quiz questions correctly so far and love seeing the mouse points. Might just be me. Yeah, the second lecture on Monday, I did it at home because I didn't feel well. And so I get to use the mouse, which doesn't happen in all the other lectures. Let's look at this. This is typical. Elise writes, RFK Jr. in summary stated he never said certain chemicals in our drinking water cause boys to become girls. Even after they showed his video stating that, he denied it. So it's just a fantasy world. I don't get this. This is like what you thought would never happen. Can you remind us why inactivated poliovirus cannot be used in an oral vaccine? I believe it just doesn't work as an oral vaccine. I mean, there may be some kind of inactivated vaccine that you could give orally and that would work, but IPV is not it. Hello, Joseph from Ontario. Polio Pete, hello, welcome back. Hopefully the RFK5N1 virus won't spread. He should be committed, not confirmed. Yeah, as James writes, infectious disease info might not even flow to us. That's correct. Aspie writes, WHO without the world is just an HO. That's very funny. Sabrina, good evening, everyone. Very windy day. Took my heat out. Strange times in the States. Wish I was in Italy. Yeah. Italy's got its own problems. Nothing stops local and private agencies from reporting to the WHO. I bet some data will still flow. Yeah, maybe, under the table stuff, people who know people. You just don't know the veracity of it. That's the problem. Abdur Rahman, the first virology lecture is a lot of nostalgia. Thank you. Did he innocently get Medicare and Medicaid confused, or did he really think Medicaid has premiums? Yeah, he's confused. He doesn't do any homework. He probably didn't read anything. He didn't prepare at all for these hearings. The words cowardly. Yeah. They are cowardly. Hello, Truth or Dare. Welcome. Angry Penguin writes, I saw in one of your lectures that some viruses attack fungi. Can you please talk briefly about one? Yes, I could. There's a... Yeah, let me get something ready for you next time. I'll do that. There are lots of good examples, yeah. Frank, HHS needs an expert as HHS secretary and the head of CDC, not politicians. I vote you and your entire team take over HHS and CDC. At least we'd be informed. Well, I'm not qualified to run any of these agencies. They're too big. I don't have any experience in it. No, but Daniel could, because he's run a big group of doctors. And right, MN, science gonna science. It's true. Hello, NY Debs from upstate New York. JH wants to know, should we be worried about our outdoor cat bringing H5N1 into the house? Well, I don't think there's much you can do about being out on fur and paws, right? But just don't let it bring any bird carcasses in, okay? That would be a high load of viral load. Hello, Mood Indigo in Bangalore, 75F. Lise writes, Sanders and Warren held RFK Jr. feet to the fire and he just could not wiggle out of it. I love when people are really good at that. And it's so obvious that the person being questioned knows nothing. Does anyone else die a little inside when people start going on about bird flu? Oh, I don't. I think we're on top of it. I think we have vaccines and antivirals we're monitoring extensively. I'm not too scared about it. What I'm scared about is if RFK Jr. is confirmed, he might not allow any work to go on, any vaccine work, for example. Trine wants to know if you stop polio vaccination, what's the lag to poliomyelitis to become great again? Probably a couple of years. Because, like in the U.S., the virus is circulating and you just need to get enough non-immune people. As kids are born, they're not going to be vaccinated. That'd take a couple of years to build up enough kids. Laurie writes, I'm afraid RFK Jr.'s new Lysenkoism will kill a lot of people. Rita writes, I want to take health advice from someone who, through his eating habits, infected himself with a brain worm. Yeah. Of course, it's ridiculous. He doesn't believe in germ theory of disease. Did anyone ask him about that? How can you say you don't believe in it when it's clear that thousands of experts have proven it to be true? Opioids is a major. What he meant is he took a lot of drugs. Mood writes, did anyone talk about the bear dump in Central Park? Isn't that some sort of health hazard? I don't know. Did anyone talk about that who listened? Visto. HPV vaccine doing well in Australia. Cancer rates are falling. Vaccines come from the U.S. I'm worried about the future. Yeah, you're worried about whether he can prevent its manufacture. Well, the companies like Merck, they will sue to be able to manufacture it. You can't stop them from doing that. What he can do is say, repeat all the placebo-controlled clinical trials, which for many diseases you can't do because there's not enough disease. And so then he'll say, then you can't use that vaccine. This is just nonsense. Just illogic. But I don't understand, what's his end game? He just wants to kill a lot of people? Are there tests for Nova virus? You mean norovirus? I think there are, but let's take a look. Yeah, but it's not not an over-the-counter. It's a PCR test, so you can't do it yourself. Muhammad wants to know any new evidence about fomite viral transmission. Not particularly. We know for some viruses it's a big deal, like rhinoviruses and noroviruses. Fomites can play a big role in transmission. Spamlet just did some science. I made a paste of tapioca starch to suspend poppy seeds on my bread. I tried to sweeten it with honey and it went watery. The honeybee has bee spit amylase in it. Yeah, that's cool. Isn't that nice? In the hearing, Susan writes, in the hearing today, RFK Jr. really only claimed to agree with the polio vaccine and the measles vaccine. Well, he's changed his mind about those. But, all the others work very well. HPV is a great vaccine. Yeah. Will writes, Shen Kwai Le. Happy New Year. Is it Happy Lunar New Year right now? Thank you. Same to you. Sabrina writes, it's an alternate universe administration. So much misinformation to intentionally confuse people and cause chaos. They don't care about the damage they do. Yeah, but I don't know why you'd want to hurt people, especially kids. What did they do to you, right? Artemis, a friend who read the Tamiflu monograph, concluded that when you get on average half a day less of symptoms, but during the period you have them, they are worse than without Tamiflu. Yeah. The other antiviral, baloxavir, is much better. I got to sit up. I'm slouching. Tony writes, there were a couple of senators who did give RFK a hard time. Were they mainly Democrats, I would guess? Yeah. Hello, Richard from El Segundo. Barely survived the hearing. Will USA exit from WHO will mean WHO funding goes down 20%. That means death in other parts of the world. Heather writes, Debunk the Funk did a great video today about the stopping of PrEP. Heartbreaking what it might do. It's just meanness. Just pure meanness. Rad was listening to Vinay Prasad's comments on the CIA lab leak report. And he also mentioned the 12 episodes, how boring and wrong the episodes are. It's disappointing to hear such rhetoric. Yeah, sure. We're wrong. We're scientists looking at the data. We're not wrong. You're the one who's wrong. But it doesn't do any good to say you're wrong back and forth, Vinay. You have to say exactly what is wrong with our analysis of the papers. Now, today they resumed the PrEP in Africa. Good. That's very good. In his case, boring means he can't understand it. I often pause Twiv to look things up. Why does Africa need the U.S. to pay? Because they can't. They can't afford it. And we can. We're a wealthy nation. We can help other nations with their health care. No one is safe until everyone is safe. For the White House, now gender is assigned as determined at conception. Good evening, ladies. Yeah, it's crazy stuff. Yeah, RFK is not a serious person. He shouldn't be treated and he shouldn't be nominated for this position. But he has been, so we'll see. Farhad, my girlfriend found out she didn't take Hep A vaccine. Her PPO insurance doesn't cover it, even though it's preventive care. What's the logic behind it other than lowering the cost? There's no logic. Yeah, they're cheapskates. The health insurance companies try and keep as much money for themselves. It's disgusting that everybody's motivated by money and not by your health or well-being. Adam writes, have you heard about clinical trials for long COVID using HIV antivirals? I think I have. Let's see. Yes. Trial of repurposed HIV, Truvada and Maravirac. It says they can reduce symptom burdens in patients with long COVID. I think that's great. I don't know what the mechanism is. It's interesting, but it's good, right? Anything can help. Laura writes, if WHO reformulates the upcoming year's flu vaccine, can the pharmaceutical companies tell the USA? Or is it a moot point? Will there even be a flu vaccine in the fall? Well, I think there has to be a flu vaccine in the fall. I'm thinking. Yeah, the WHO committee makes the recommendation, but there are U.S. members on it, but they're going to be pulled off. But that can be known from other countries, so I'm not too worried about that, the composition of the flu vaccine. There's another reason why it's dumb to drop out of WHO. And also someone wrote, I forgot to highlight it, it's a good part of the budget the USA pays, and that's going to hurt WHO. They don't care. Yeah, look at Costello. Why is your cat even outside? Yeah, I think it's dangerous being outside as a cat. I mean, as a kid, I had a cat who lived outside and he got beaten up all the time in fights and eventually died. Hmm. Hmm. Peter says, unfortunately, I'm afraid the vote on RFK will go the way of the Secretary of Defense, 50-50 with a VP tiebreaker. All politics, no science. It's ridiculous that these people are hearing this crap that he's promulgating and they're still going to vote for him to appease Trump. It's horrible. This can only lead to disaster. Hmm. Abdur Rahman says, as an MD, we are inclined to withhold care to most of these politicians. Maybe they can write a law that says they're healthy or changes the definition of disease while decomposing. Can't do that as an MD. You're sworn to take care of everyone, right? Lise writes, State Department reinstated HIV medications for worldwide treatment for times today. It's crazy that we have to be at this point, right? Patricia says, lots of dead birds being collected in Massachusetts. It's concerning. Yeah, it is. John writes, maybe I'm naive, but I think it would be symbolic to vote him down, even if they parade out more unfit candidates after him. I don't know why Murkowski and the other lady can't vote against him. They seem to, the old guard Republicans seem to have some independence. Maybe they're afraid of Trump. Yeah, Patricia says, I'm not sure anyone asked about germ theory. Yeah, do you believe in germ theory? I don't. Why not? What's the evidence that it doesn't exist? My God. Polio Pete writes, is SARS-CoV-2 the first RNA virus that a vaccine was developed? Why not other RNA viruses? So there was an experimental vaccine being developed for, RSV, which eventually got developed as a protein form. But yes, SARS-CoV-2 were the first mRNA vaccines. Mm-hmm. Why not other RNA viruses? Well, we had a lot of already existing vaccines that were very good. And, there were issues with getting the RNA vaccines to work, which were solved not too long before COVID, and then we used them right away. So we didn't have really time to apply them to other viruses. Richard says they did ask him about germ theory. He was evasive. Hey, they should play a clip saying, I don't really believe germ theory. That way of talking he blames on the flu vaccine, by the way. And this has nothing to do with anything, and I shouldn't mention it, but I can't stand that he clearly washes his shirts and doesn't bother to iron them. Just pulls them out of the dryer and they're all crumply. He just looks like a kid who can't dress. Oh my God. It doesn't matter, but just weird. RFK also lied about where he was living. If someone could lie for something so little, it means that person is not trustworthy. Jolie Moly from Portland. Hello and welcome to this crazy world. Spamlet writes, when chicken barns get flu infected, instead of culling, would it be better to wait to check for birds with immunity? No, it turns out you will just spread through the herd or the flock even more rapidly. So that's why they do that, and that works very well. Polio Pete, maybe VR will address this. For start, polio is an RNA virus, flu, measles. Yeah, so they had reasonable vaccines before COVID. And since they have been tested, I know that flu has been tested as an mRNA vaccine. It doesn't really work. I don't know about measles and polio. You're the wood snake. All right. Elizabeth Parker writes, the WHO's data on flu will be public, and most vaccine manufacturers work worldwide. There will be vaccines. The issue will be approvals in the U.S. and paying for people to get it. Thank you. Thank you. John wants to know, can you give a definition of convergent evolution? Yeah, you have a property that you find in two different viruses, like herpes virus and poliovirus. And you trace that property back and you find that it existed in ancestors of each virus. So it evolved independently towards the same function. You can go back and show that, in each lineage, that function was slightly different, but the convergent evolution, the selection pressure ended up giving you the same product after so many years, right? Get the same thing. Cats are hunters. Adopted cats from the outdoors are impossible to keep inside and keep happy. I know. It's hard. Lise took my car to the car wash because I parked under a tree. Birds pooped on it. Car wash didn't do a good job, so I did after. I thought probably not a good idea with bird flu. Well, you could wear a mask and you'd be okay, right? So if you do it again, just wear a mask. Aspie writes, I have 2 rescued ferals right off the parking lot. Those cats won't ever try to go back at there again. Costello, the adopted cats from outside that moved in here are absolutely convinced that outside shouldn't exist. Straight offensive. That's great. China said they will take over WHO funding. We just are throwing ourselves on a sword. Yeah. They should. It's great for them. Has anyone mentioned the suspension of the weekly MMR, emerging infectious diseases? Yeah, it's horrible that they're suspended. One hopes that they resume. I think they will. They have to, because those are important publications. Other vital statistics, any kinds of communications about anything like this product has an issue, right? This sausage is contaminated. They're not going to issue those for a while. Yes, this week in feline behaviors. Every week we get on some tangent. This week it's cats being feral cats, whether they like to stay outside or not. Oh, 250 people here tonight. Wow. Come for the science. Stay for the chat. Happy Lunar New Year. Ray, what are the differences between a bacterial infection and a viral infection? Well, they can be acquired in similar ways. They can be like respiratory bacterial infections, intestinal bacterial infections, blood infections. Viruses can do that as well. Bacteria tend to cause skin infections. There are some viruses, obviously, that cause skin rashes and so forth, but I think the bacteria can cause the really necrotizing skin infections. But basically, bacteria divide. One bacteria becomes 2 and 4 and 8 and 16. And we have lots of antibiotics to treat them. Whereas viruses need to get into a cell. Bacteria do not need to get into a cell for the most part, although there are some intracellular bacteria. And viruses need to get into cells. So they can both be nasty. You don't want either. You can get bacterial infections from other people. Same with viruses. You can get them from the environment. Same with viruses. So in many ways, they're similar, although biologically there are differences as well. Scott writes, people travel the world and diseases travel with them. Wealthy countries are helping protect themselves when protecting others. It's cheap protection when compared to treatment and worse outcomes. Sex-wise, isn't the dice cast as soon as the spermatozoa enters the egg? Apparently, it's subtle, and I don't really appreciate the science there. Maybe someone else does. Yes, antibiotics won't touch a viral infection. That's right. And antivirals won't touch a bacterial infection. Will says health and well-being. Robert Reich wrote a brilliant book, The Common Good. Yes, you recommended that. I have to get it. Which has been replaced by whatever it takes to get a profit. Thank you, Scott, for your support of science communication. Really appreciate it. RFK is just one of those people with a well-founded inferiority complex who will assert nonsense, make themselves feel more important. You probably saw Caroline Kennedy. She made a video and wrote a letter saying, I don't normally comment on my family's issues. We have a complicated family, but this guy is not qualified to be head of HHS. He just knows no science. He's not interested in health. Unfortunately, they're not going to listen to her. I think this is really good. They're going to pick up our contribution. Like, let the U.S. be petty. That's what we are. Silliness. Trump thinks he can just play these games, and get what he wants. Yeah, I understand the average lifespan for an indoor cat versus outdoor is a big difference. Yeah. Yep. Drolly, maybe the U.S. should set up its own international aid organization. Less bureaucratic, less political, and more Africa focused. Well, you have some like Doctors Without Borders, Parasites Without Borders, and others, but they're relatively small. Although Doctors Without Borders is large. It's not a U.S.-based one, but I think those are good. Nonprofits can do that, and I don't know if they could stop that. They could raise money to do it. Yeah, I think it's a good idea. Regarding bird flu, I'm wondering how safe it is to hang laundry out on a line. Well, you could hang it out and immediately a bird can poop on it, right? So there's no time limit. It's just when the birds are going to be there. Vexing cat. Thank you for your contribution to science communication. Peter writes, long COVID in long COVID, virus persistence has been determined by PCR. Is it replicating virus or viral debris? It doesn't get eliminated. In my opinion, there's no evidence that it is replicating virus. It's very hard to show, no doubt, but I would favor its persisting pieces of RNA. Yeah. Do you think it's realistic to expect politicians to understand science? We've never chosen politicians with science qualification. That's a shame. There have been a few, right? But yes, you're right in general. Although, Elizabeth Warren wrote this great letter about RFK. Obviously, she has a staff that does great research, okay? Because it was very knowledgeable and really well referenced. You can find it. But it's a problem, right? Yeah. And this is an example of that. Even though you say he's wrong, they say, we're going to vote for him anyway. It's politics. They don't care about the science. It's all about giving Trump what he wants. Scripps and UW dug up in sequence 1918 flu. When should we expect it to drop in Trump's presidency? Well, it's already been done by others. So I don't know that they can make a big deal about it. So I don't think so. Although they might, just to set an example of scientists doing dangerous experiments, right? That's what they like to talk about. Kang writes, sorry to be late, but this administration doesn't think an ounce of prevention is worth a pound of cure, and they are not looking ahead, but only looking to settle odds. Old grudges. Yeah. All right, I'm gonna mark that. I'm gonna do a mini lecture, but first I want to go all the way back to the beginning. So here's Caroline Kennedy's video about her cousin. And Questella writes, Vincent has meant more to me than any teacher I ever had in real life. So that's very nice. That's very kind of you. If other teachers gave half the shits Vincent does, society would be a million times better off. Thank you very much. Appreciate it. Okay, let's do some. So a mini lecture. It's not going to take very long. Let's talk about viral RNA, because we're always talking about RNA. But what is it? And how does it work? So, there's RNA and there's DNA, and they're remarkably chemically similar. So, DNA and RNA is a long chain, really long, right? DNA is very long and RNA not as long, which is basically made up of sugars linked to each other by phosphate bonds. So here's a sugar, this ring here, it's a ribose. And here's another one. And then there's the phosphate that is in between the two of them. And this goes on for a long time. And then there are bases attached to the sugar. And so the bases, like here, this is guanine. There can be any base there. So the bases in DNA are A, G, C, and T. And in RNA, it's A, G, C, U. And they can pair with each other. So that's how you make double-stranded nucleic acid. So you'll have one strand with bases. And if this is a guanine, then it will pair with a cytosine on the other strand. And those are the rules for base pairing. Right? And so see this hydroxyl here? So this is RNA. If you replace this OH with an H, you have DNA. And that's a difference between the two. And also, one of the bases is different. So in DNA, it's a T or a thymidine, and in RNA, it's a uracil. So they're remarkably similar, although structurally they look very different, and RNA is very fragile and so forth. But this is RNA versus DNA, okay? And it's this little hydroxyl. So ribonucleic acid, so ribo refers to the sugar, and deoxyribo means we're taking off this hydroxyl. Deoxy, it's just a hydroxyl, and that's DNA. That's very cool. So RNA history. Remember, tobacco mosaic virus was the first virus to be discovered. In 1935, it was crystallized by Stanley, and he found that the crystals have 5% RNA. But he thought it was a contaminant. He thought the protein was the genetic material. But in 1944, DNA was shown to be the genetic material of bacteria. And in 52, the Hershey-Chase experiment showed that DNA is the genetic material of some viruses. The DNA structure was solved in 53. In 56, Frankel and Conrad showed that for tobacco mosaic virus, the RNA is the genetic material. And by 1959, RNA was identified in many animal viruses. So it was unusual. Stanley was wrong. In fact, the RNA was the genetic material. He got a Nobel Prize for crystallizing this thing and saying that the protein was the genetic material. It should have been taken away because he was wrong. So in the Baltimore scheme, which I've shown you before, right, there's 7 classes of nucleic acids. And the RNA viruses are shown here. Double-stranded RNA, negative strand RNA, plus strand RNA, and plus strand RNA with DNA intermediate, the retroviruses. So all those viruses have RNA in their genome as their genomes. So how do you copy this RNA? So in the 60s, an experiment was done actually by David Baltimore. Where he took cells, he infected them with poliovirus, and then he broke them open and added the 3 triphosphates, which are the building blocks of RNA, right? A, U, G, and C. And one of these was radioactively labeled. So then he measured RNA synthesis. And at the same time, he kept some cells intact, and he did a poliovirus growth curve. So you can see here, poliovirus PFU per mil in red. So you can see a little eclipse period, and then poliovirus production begins. But in these cell extracts, you can see RNA synthesis begins about the same time that virus particles are made. And so that's the first indication that in virus-infected cells, there's some kind of RNA polymerase activity. And subsequently, so that was a plus-strand RNA virus, right? Poliovirus. And then he said, what about negative strand viruses? Because he reasoned that if you're a negative strand, when you get into a cell, you can't be translated because it's a negative strand. So he said there has to be a polymerase in the particle to make a plus strand, an mRNA. And in fact, he looked in VSV and he found a polymerase in these particles. And so that's where we are today. We can see in all RNA viruses, they encode an RNA polymerase. Plus strand viruses, they don't carry them in to the cell, and in minus strand viruses, they do. So here's some examples of RNA and RNA-dependent RNA polymerase in the virus particle. That's the enzyme that copies RNA. Only viruses have that enzyme. So minus strand genomes, they have to have the polymerase in the particle, and the RNA is also coated with protein. It's called the nucleocapsid. So like VSV and influenza virus, the RNA polymerase is in the particle. The plus-stranded RNA viruses, there's no polymerase in the particle. And for the most part, the RNA is naked except for retroviruses and coronaviruses. And then we have our double-stranded RNA genomes. They have an RNA polymerase in them because the double-stranded RNA can't be translated by ribosomes. So the polymerase has to make mRNA from these double strands. So nucleocapsids are protein RNA structures, and we've solved many of them. Here, for example, is a schematic of the VSV nucleocapsid. You have the RNA there in green, and then multiple copies of nucleocapsid protein wrapped around the RNA. Then on the bottom is a molecular view. This is the structure of the nucleocapsid protein. And on the left, the structure of many copies of it bound to the RNA. That dark part, which you can barely see, is one molecule of the nucleoprotein. So on the right is influenza virus. Again, the genome in 8 RNAs, negative strand RNAs, is coated with protein. And here you can see a schematic of that. It also has the polymerase attached to it. Here's a single molecule of the nucleoprotein, and here it is attached to the RNA, 2 different views. Those are nucleocapsids. Now, RNA structure is not a line. I often show lines in class, but they're actually highly structured RNAs because they have these base pairs, these internal base pairs, or stem loops, as we call them, where the RNA can base pair with itself. And they can be quite extensive, actually. Here's one for HIV. And you can see its structure is quite involved. And these structures are important for RNA synthesis. So here's a molecule of RNA. And the rules for RNA synthesis is that if this is the genome, it has to be copied end to end. You can't lose any nucleotide sequence, right? Then you can't make a virus. And you have to make mRNAs that can be translated by the cellular protein synthesis machinery. Right? Those are the 2 main rules. And so here's a model of how RNA synthesis occurs. Here's a template that's going to be copied. And here's a polymerase using a primer to make a copy. So synthesis initiates and terminates at very specific sites. Some need a primer. Some polymerases need a primer. Others don't. We usually need other cell proteins and viral proteins. And the way it works is that you stepwise incorporate NTPs into the growing chain depending on what's in the template. If there's a G, you put a C in. If there's a U, you put an A in. Although there is some non-templated synthesis that doesn't depend on copying bases. So here's the way catalysis works. Now this may be a little bit weedy for most people, but I think it's very interesting. This is actually DNA, but it applies to RNA as well. It's called the 2-metal mechanism of polymerase catalysis. So again, we have a polymerase using a primer to add bases to a template. And on the right is how it happens. So here's the template strand on the right. So there we have a G attached to a sugar, a T attached to a sugar, an A, a T. And here's the growing strand. So we've added a C and an A, and you can see there's a phosphate bond joining them. So these bases that are used for synthesis, like ATP, they have to begin with 3 phosphates. And in the polymerization, we take 2 off. That provides the energy for making the bond. And so that's shown here, actually. So we're adding this 3rd base, T, which is base pairing with A. All right, so it's a little complicated. Here's the sugar, and it's got attached to it 1, 2, 3 phosphates. Okay, And you see here, there's a deoxy. So there's an hydrogen here. There's no hydroxyl. So what happens is basically there's a series of attacks, chemical attacks, that basically break off phosphates 2 and 3 and allow phosphate 1 to hook the two bases together. And the energy liberated by these allows for the formation of that base. So that's how DNA and RNA polymerase catalysis occurs, and it's really fast. There's some great movies on YouTube that you can go and watch and see this. Now, the enzymes that do this are very interesting. There are 4 different kinds of enzymes. So there's RNA polymerases like poliovirus here. There's the reverse transcriptase like HIV. There is an DNA-dependent RNA polymerase. And then there's a DNA-dependent DNA polymerase. These are 4 different enzymes. They underwent convergent evolution from some common ancestor. Right? So they were a common ancestor, and they split into 4 lineages at some point, and they evolved in a similar way to give a similar structure. And these enzymes have common areas. The most important one is this yellow area, which is where the catalysis occurs, where the bases are added. And I'm going to show you some detail for the polio polymerase. So here it is in a little more detail. So these polymerases are referred to as a right hand with thumb and fingers domains, and in many cases they're touching, like the polio polymerase, the fingers and thumb domains touch, which gives you a closed active center. And then there's the active center where the catalysis happens. It's red here on this hand. It's yellow, these 2 beta strands in yellow. That's where the enzyme does its job. It makes the RNA strand, basically. It's very cool. And these are structures to show you how this works. So here's a top view of the polio polymerase. And the template goes in the top. It makes a right-hand turn, goes past the active site, which you can just barely see in yellow. A second strand is added, and then it comes out the exit channel. Here's a front view, same thing. The RNA template goes in, makes a right-hand turn, then comes out double-stranded. And in the back of the molecule is the NTP entry. So the nucleic acid is coming in and out in the front, and in the back, the NTPs are coming in to help the polymerization. This happens really quickly. And you may ask, well, how does the enzyme know which NTP to bring in? It actually doesn't. It brings them all in, all 4 at once. It says, take your pick. It tries A, G, C, U, and whichever one works is added, and then it moves on. Because there's no way to know which one to grab from the environment and put in there. It's a very cool process. Now, for plus-strand viruses, two kind of general ways that RNA are made. So here are the picornaviruses and the flaviviruses. Their genome is a single strand of plus RNA. Of course, that can be translated as soon as it gets into the cell, right? It's copied into a minus strand in order to make more plus strands. And then some of those can be used to make protein. There are some plus strand RNA viruses called alpha viruses, like chikungunya virus. They have a plus strand RNA, and they make a minus strand intermediate, but they also make a subgenomic mRNA. It's just a small mRNA rather than the whole genome. It's a different way of accessing the coding region. And so for poliovirus, the way it looks is the virus binds a receptor, comes into the cell, the RNA gets in the cytosol, it's translated into proteins. Among those proteins are the polymerase, which then start to copy the plus RNA into minus and back into plus. And eventually those RNAs are used to assemble new virus particles from the proteins that have been made. So that's a quick overview. But here on these membranes, that's where the RNA polymerase is churning out viral RNAs. Now, coronaviruses I put in here because it's relevant, obviously. Very complicated. The genome is 30 kb in length. And once the RNA is in the cytoplasm, the first half is translated, and this sets up vesicles where RNA synthesis occurs. And that kind of RNA synthesis involves genome replication and the synthesis of mRNAs. And so the genomes are made from minor strand intermediates. They are coated with nucleocapsid protein, and then they acquire a membrane intracellularly. This nucleocapsid RNA structure buds into this compartment, the ER-Golgi intermediate compartment, and then that's sent to the surface by vesicular transport. So that you may be wondering how you get translation of the rest of this genome. Well, you make a series of mRNAs that encode for the proteins on the right-hand half of that genome, and you can see them here encoding a variety of proteins here, for example, the nucleocapsid protein and the spike protein and so forth. So it's a really complicated system. And in these mRNAs, they each encode a lot of proteins, but only the first one is translated. All right, so here the spike is the first open reading frame. Then all the other open reading frames are not translated. Minus strand RNA viruses, of course, the minus strand cannot be translated. So the virus particles have to have an RNA polymerase in them so that when the virus infects the cell, it can make mRNAs to get things going. And these viruses can be what we call unimolecular, one negative strand RNA like VSV or rabies virus, or they could be segmented like influenza virus. And the general scheme is the negative strand RNA comes into the cell. The polymerase attached to it makes mRNAs. Those mRNAs are translated to proteins, and they drive the assembly of new virus particles. So here's a scheme for the replication of VSV mRNA. So again, the VSV genome is released into the cytosol. It's negative stranded, so it can't be translated, but it has a polymerase attached to it, and that makes mRNAs, individual mRNAs that encode viral proteins that can drive synthesis of RNA and also assembly of new virus particles. And there's a key aspect here that I want to tell you about. That is, when the viral genome is not messenger RNA, which means it's minus, there must be a switch from mRNA to genome RNA synthesis. So here you see we're making mRNAs. But you can't just keep making mRNAs. You need to make genomes so you can make virus particles. So at some point, you have to have a switch. And we know what that switch is, actually. So here's the genome of VSV, right? Bullet-shaped particle, encodes 5 subgenomic mRNAs. And essentially, when the negative strand RNA gets into a cell, the polymerase attached to it makes mRNAs. When the level of N protein, the nucleocapsid protein, gets high enough so that it coats all of the mRNAs, you now make full-length plus strands. And so the switch is controlled by the level of N protein. So early on, there's not a lot of N protein. You make mRNAs. Later on, the N protein coats the mRNAs, and it becomes a full-length plus strand, which is then copied to a minus strand, and that's how you make new virus particles. So they're very clever. Evolution is amazing. Now, influenza is the same thing. The genome is not mRNA. The complication here is that there are 8 segments. It's a little bit complicated. And again, there's a switch from mRNA to genome synthesis. The additional complicating factor for flu is that the RNA synthesis happens in the nucleus. So the genome segments get in the nucleus, They're made into mRNA. The mRNAs go out in the cytoplasm. They're translated. Some of the proteins go back in, and eventually you switch to full-length plus strand synthesis, just like for VSV. So here's the flu genome. Each segment encodes an mRNA or two and one or two proteins. And again, the genome comes in. It's translated to make mRNAs. It's copied to make mRNAs. They're made into proteins. And then when the levels of nucleoprotein get high enough, they coat the plus strand, and you get full-length plus strands from which you can make new minus strand genomes. Now, the primer for mRNA synthesis is actually a piece of cellular mRNAs. And so there's an endonuclease in the virus particle that cleaves mRNAs, and those are used to prime synthesis of plus strand RNA. The endonuclease is the target of the influenza inhibitor Xofluza, which is supposedly better than Tamiflu. You need just one dose, and you don't need to take it within 24 to 48 hours, and apparently works really well. So think about that next time you get flu. Ask your doc for Xofluza. This endonuclease was discovered by Robert Krug, a virologist who I've known for years. And he wrote into Daniel some time ago, "Why don't you guys prescribe Xofluza? It's much better." And Daniel said, "I don't know. I just never thought of it." It's all about the companies pushing their own product, you know. All right, and finally, double-stranded RNA viruses, like reoviruses and rotaviruses. So they have double-stranded RNA in the virus particle, but it can't be translated directly. So there's a polymerase in the particle that makes mRNAs from the double strand, and you can then make protein. You can also take some of those mRNAs and copy them and make double-stranded RNAs for making new particles. And I want to show you the reproduction cycle of this virus. It's very cool, right? So it's reovirus, multi-segments of double-stranded RNA. The virus attaches to a receptor. It's taken up into the cell by endocytosis. As the pH of the endosome drops, the outer shell of the virus comes off. This virus has two shells, and that makes the virus hydrophobic, and it can poke through the endosome into the cytoplasm. But it still has the double-stranded RNAs in them. They are copied right in the particle. The mRNAs come out of these five-fold turrets, and they're translated into proteins, and they can be used to build new capsids with single-stranded RNAs and then double-stranded RNAs. So it's very cool that the mRNAs are made in the particle, and the mRNAs come out of these turrets. So here's the virus particle and the progression to the core that pokes its way out of the endosome, because it's these portions that stick up are hydrophobic, and they're covered up in the double membrane or the double capsid particles. Very cool. Also, there's a plug here at the five-fold turrets. They come out, and the mRNAs can come out of those channels. You gotta love virology, don't you? So here's a picture of the release of mRNA from rotaviruses. So here's a particle undergoing mRNA synthesis. The mRNAs are coming out of the five-fold axis, and each double-stranded RNA segment is attached to the polymerase, which is underneath the five-fold axis of symmetry. So you got polymerization happening at each five-fold axis. Very cool. And finally, RNA viruses make a lot of mutations, far more than DNA viruses. Their polymerase make errors, but there's no proofreading activity, and they can make a mistake in every 1,000 to 10,000 bases polymerized. And so in a 10 kb virus genome, a mutation frequency of 1 in 10 to the 4 gives you one mutation per genome. You make 10,000 genomes in a cell, and you've mutated every base. So that's why it's silly for people to say the virus is mutating. It always mutates. Now, the exception are the nidovirales. They have, error correction. It corrects polymerase errors. And you can remove this exonuclease, it's called XON, and it will give a virus with a 15 to 20-fold increase in mutation rate. And that's probably why, in part, we can get large nidovirus genomes, because they have this error correction protein. Love it. RNA viruses can also recombine. This happens among plus-stranded RNA viruses. And basically, the polymerase is copying one RNA, and it can then start copying a second if the cell happens to have a different RNA in it as well from a related virus. And this is relatively high. So for poliovirus, 10 to 20% of poliovirus RNA molecules recombine in a single replication cycle. So it's very high frequency. It's why the NOPV2 lost its changes that were so painstakingly introduced by 2 recombination events. And then it was just another poliovirus causing paralysis. Any questions? So now you know a little bit about RNA. And I like to teach, yes. I do like teaching very much. So let's go back down to the end of your questions, because there are more. I started where I stopped. So this time I got smart to some extent. So now I have to find out where I stopped. Did I stop here? I must have been, yeah. So I stopped with John's contribution. Thank you very much. Now where did it go? Here we go. Okay. I am not drunk says RFK Jr. is one of those people with a well-founded inferiority complex who will assert nonsense to make themselves feel more important. I hate when there are personality syndromes involved, right? With this, it's just too bad. Yeah, Elizabeth Parker says, not necessarily. It's not just about the chromosomes. Hormones during development can change the baby's sex. It's complicated. That's what I thought. Yeah. Wow, Will has 9 cats. That's cool. I think I already answered those. Vexing cat. Yes, thank you for your contribution. Yes, we talked about that one. H5 from Visto, decimating chooks and Australian egg production. It's possible to predict what form of recombinant flu that infects humans will look like. No, I think we don't know. It has to have alpha-2, 6 sialic acid binding. It has to have a change in the polymerase to allow it to grow effectively in mammalian cells. But then it needs changes that allow it to transmit from human to human, which right now it can't do. And we've not ever seen that happen. Of course, we've never seen so many infections of animals. So maybe it can happen, but nobody knows. Nobody knows. You just need to be prepared. What do they do with cold birds? Well, we heard from a guy who works in a facility. And what they do, he said they grind them up and they bury it. They bury the ground up birds. And he was concerned that he was going to get infected, right? Because this ground up chicken is going in his face, even though he has mask protection. So that's what they do with them. Tip for egg price concerns. Raw scrambled eggs in airtight containers will keep in the freezer for up to a year. Wow, so you can stock up. Hello, Steve. Happy birthday, Steve. You young guy. What do we know about HPAI's persistence in water? Yeah, I mean, it infects aquatic birds, and they poop it out, and it goes in the water, and other birds pick it up, so it persists very well. Yeah. Hmm. Yeah. Well, they're ground up. This guy said they were just buried, because if you burn them, then you're going to make an aerosol of all that. So I'm not sure that's good, but probably it's a number of different ways. Yeah. What do you think could happen to Dr. Fauci? Well, the crazies everywhere in the world. He should lay low, because people just shooting people left and right now with no regard, because they'll just get pardoned by Trump. Yeah, RFK says he'll keep making money on anti-vaccine lawsuits when he's in office. It used to be that you couldn't do this kind of stuff, but all rules are out. These guys are like, I can do whatever I want. It's just, it was just an agreement that you would behave a certain way, and it's out the window. Now explain capsid versus cell wall versus mycoplasma. So the capsid is the protein structure surrounding the virus particle. So I think we had a capsid in the lecture. Let's see. So here, for example, is a reovirus capsid. It's a protein shell that protects the genome. A cell wall is what surrounds some bacteria. Okay. And it's made of peptidoglycan, and gives the bacteria a lot of rigidity and structure. Now, mycoplasma are organisms, prokaryotic organisms. They are not bacteria, but they are prokaryotic. Let's get a good definition of mycoplasma. Genus of bacteria that lack a cell wall. Okay, and so many bacteria have cell walls. Let's get a picture of a cell wall of a bacteria here. See if I can find a good one. This is not a bad one. It's too small though. All right, so here, for example, is a bacterium, and we have the cell wall on the outside. It's very thick, and then below that is the plasma membrane. So the cell wall is yellow there, not so thick. The capsule, which can be present, is thick. Cell wall, peptidoglycan, and then the plasma membrane. And then mycoplasma are bacteria without cell walls. Good question. Yeah, I could stream to Twitch next week if I can remember. Let me make a note. I'm really bad at remembering stuff. Go to Twitch. I probably wrote this down already a million times, and I just pile up my stickies. Twitch. Yeah, I think I just push a button and we go to Twitch as well as this place. Okay. Pili. Explain bacterial pili. Here's the same picture. The pili are these protrusions from the bacterial cell wall or capsule. These are ways that many viruses can attach to these pili and infect the bacteria. And also DNA can get into the pili. So they're evolutionarily very important because they're a means of exchanging genetic information. Plasmid transmission. Right. So plasmids are circular DNAs that can reproduce in bacteria, and they can move from bacteria to bacteria in different ways through viruses, but also through these pili. Yeah. Thank you, Christella, for your support of science communication. Really appreciate it. Oh, 183 people, 199 likes. Thanks, guys. Great job. Why didn't anyone on the panel ask what school of science did he go to? Well, he didn't, so they didn't want to embarrass him, but I think that's what you need to do. Frank said a pigeon pooped on me yesterday in Manhattan. Yeah, it happens. He'll be okay. Mystery Fine, thank you for your contribution to science communication. Really appreciate it. Sabrina says, I've said for years that there should be more people from science and humanities backgrounds in the Senate instead of career politicians, lawyers, and business people. Yeah, but scientists want to do science, right? It's a problem. And then maybe you're a scientist, you go into Congress and you become a politician. You forget your science. So there have been some scientists and they've been good advocates, right? But I can't name them for you. Angry Penguin says, in the early 90s, our company CEO said we sold our IPV to some African countries at 7 cents per dose because that's what they could afford. Wealthy countries made up the profit shortfall. That's very good. I like that. That's the way it should be. With the high cases of bird flu, what's the probability it will infect humans? Well, as you know, we've had quite a few cases among dairy workers, right? They get the bovine version, which is mild, well over 100, I believe. And then there's the more virulent avian form, but there are fewer of those. Well, the more people it infects, the more possibility is that it could change to transmit. But I can't give you a number because I would be lying to you. I just don't know. Maybe coronas trigger some sort of autoimmune reaction. So there is some suggestion, but not enough data to prove it conclusively. Same for other viruses as well. If 1918 flu were to come back, would we be resistant? Yeah, it depends what it looked like. If it looked like exactly 1918, or if it had changed in some other species, it doesn't take much to be resistant, but it's possible. Yeah. Should be virus Pokemon. Got to catch them all. Right. Jeff writes, I understand how sodium chloride crystallizes, but I can't understand how one would crystallize DNA or viruses. Well, you basically make concentrated solutions of, say, the viruses with nothing else, and you use different solutions that allow the particles to align in a certain way to make a crystal. It's really an empirical procedure. And sometimes you can do this for years and not get a crystal. But there are certain conditions where the particles will line up and make like a sodium chloride crystal, except with a larger molecule, a virus. Are any viral enzymes made of RNA? Good question. Let's see. I can't think of any. I know the tetrahymena has self-splicing RNA. Of course, the ribosome, the catalysis is carried out by RNA. Some viroids have hammerhead ribozymes, right, that are self-cleaving. And I'm not aware of any naturally occurring viruses that have them. That could be wrong. Can RNA polymerases be a target for the immune system? Yeah, they can be, but they're not self, right? They're different, and they could be. But I'm not aware of any instance where either antibodies or T cells to polymerases play a role in pathogenesis. It's really a good question. This is interesting. Polymerization sounds entropically expensive, lining everything up, winding through restricted spaces. What's the cost made up of? I think, well, you use a lot of energy and you break a lot of phosphate bonds, and that provides the energy for that. Yeah. Yeah, they're viral ribozymes, but yeah, hepatitis D virus. So that's a good example that has a ribozyme in it. But there may be others. Yeah. Now, I wish we pronounced picornaviruses pico-RNA viruses. Yeah, then you could remember it, right? Does not double-stranded RNA set off alarms with the cell? Yes, and that's why the RNA stays in that shell. It never leaves, so it doesn't get sensed. It's cool, right? I wish I had some error correction. Yeah, we all do, don't we? How do the strands get exchanged during elongation? No, there's no break. The strands are close together, and the polymerase copying and simply changes to another molecule without any breaking happening. Yes. Thanks you for your support of science communication, SQL Smarty. Really appreciate it. Molnupiravir is a mutagen. Yes. No, it just raises the error rate by, it gets incorporated and it causes mistemplating. Yeah, doesn't do away with error correction. The SARS code for its own polymerase. Yes, it encodes its own RNA-dependent RNA polymerase. What are the main goals of viruses within the cells? To make more viruses. Pure and simple. I am not drunk. Thank you for your lecture. Now I am retired. I'm learning more interesting things every day than any time in my life. Unfortunately, I also forget more things, such have I taken my medication? Yeah, I'm sorry. It happens. I forget things too. Oh, this is good. Chook is domestic chicken in Australian language. Cool. Can you eat an infected bird? All right, so the problem would be, you could cook it and eliminate infectivity, but in the preparation, it's going to be raw and you could infect yourself. So you didn't even have to eat it to have a problem. Okay, folks, time to wrap this up. A lot of good questions left and a lot of good questions under the bridge. I love your questions, folks. All right. I want to thank our moderators today. We had a lot of people here today. It's great. We had Tom Steinberg. We had Les. We had Frank. We had Steph. Thank you all for coming and moderating. Appreciate it. And thanks to all of you for coming as well. You were a great audience and great questions. Very attentive and respectful. Now, the next office hours will be in a week. It's February 5th. February 5th. It's not too far off, right? Next week. Thanks, everybody, for coming. Hope to see you next week. In the meantime, please be safe. Bye-bye.